Assalamualaikum. Here I'm discussing about the gastrulation in different chordates. First of all, I'm discussing about the amphibians. Chordates have the five classes. Fishes have the just like the same as in the amphioxys. So fish and amphioxys have the same type of gastrulation. The method is the same. Amphibians included the frog, toad, and salamander. Their egg is mesolacical, that means medium moderate quantity of the yolk. Cleavage is holoblastic, that is complete. Cleavage is radial, just like the other deuterostroms, and cleavage is indeterminate, that is the fate of cell at the cleavage stage is not known. Due to the yolk, the gastrulation becomes a little bit more complex as compared to amphioxys and fishes. The small cells of animal pole, they divide more rapidly as compared to the vegetal pole because the vegetal pole has the large quantity of the yolk as compared to the animal pole. So the animal pole has a small cell and the vegetal pole have the large cell. Small cells are called the micromeres and the large are called macromeres. This type of the cell movement in case of amphibian is called epiboly, in which the cell on the animal pole they move downwards over the cell of vegetal pole. The yolk cells they form the floor of the archentron. This is the cavity formed during the gastrulation, while the notochord it formed the roof of the archentron. Notochord later on develop into vertical columns, and this is the blastula stage. These are the macromeres in the vegetal pole. These are the micromeres in the animal pole. This is the blastocele, and here is the movement of the cell through the blastopore. This is the dorsal lip of the blastopore. The cell move inward and their movement is called epiboly. As a result of gastrulation, the new cavity is formed which is called the archentron is formed. Blastoceles slightly disappear and ultimately vanish or finish. So here this is the gastrula with the ectoderm present outside endoderm, and this is called the gastrocele or the archentron because it develops into the digestive tract. This one is called the yolk plug that containing the yolk. Here this is the video that show about how the cleavages occur. First the cell divide, micromeres, macromeres, then the appearance of blastocele, then the movement of cell through the blastopore, and a new cavity that is called the archentron. It's how it develops. Now this is the gastrulation in case of reptile and bird. Their egg is same. They have the large quantity of yolk. So macrolacetal egg, antilolacetal, that means the yolk is present on one side, that is the vegetal pole. Gastrulation occur by a process called delamination, which means the splitting of blastoderm. Blastoderm is the roof of archentron. And as a result of this splitting, the two layers are formed, epiblast and hypoblast. Epiblast form the future presumptive mean future ectoderm and chorda mesoderm, that is the notochord and mesoderm, while the hypoblast it form the endoderm that later on develop into the digestive tract, and also it surrounds the yolk sac. The formation of primitive streak, here the primitive streak is formed and a Hansen node is formed. What is primitive streak? Primitive streak is a modified representative of blastopore. It is a modified form of the blastopore, just like a blastopore, and it formed within an area of blastoderm, where the mesodermal tissue is present. While the Hansen node, it is a longitudinal groove or depression that is present at the interior end of the primitive streak. Primitive streak is the area from where the cell migrate during gastrulation in the reptile and birds and also in the mammals. Here I am show you that in case of birds and reptiles, the blastodisc is formed or also called the blastoderm that split into epiblast and hypoblast. This is the blastodisc. Now it is split into epiblast and hypoblast. Here is the development of primitive streak, and once the primitive streak develop, the cell penetrate during gastrulation through this primitive streak. While in case of amphibian, the cell moves inside of the embryo through the blastopore.